Hello there, it's another short video for A-level biology. This one is about small interfering RNA. Now this topic is a little bit complicated, but it is what it is. It's a small piece of RNA that interferes with translation. So let's make sure that you're happy with the fact that DNA is transcription to make RNA, and that's translation to make protein. So let's consider small interfering RNA. You start off with a gene that is transcribed to make a long piece of RNA. That RNA folds on itself because it has complementary sequences at each end to make it double-stranded. There is a protein that we just call protein 1. Protein 1 binds to the double-stranded RNA and cuts it into small pieces. So here we will just concentrate on this middle piece. Let's get all the others out of the way. Now we introduce you to protein 2. That's all you have to call it, protein 2. Protein 2 binds to one of the two strands. The other one we get rid of, binds to one of the two strands, and that is now activated. And what that will do is look for any complementary sequence, and when it finds it, it will cut the messenger RNA that it belongs to in half. So there's that process in action. The protein and tiny short interfering RNA are scanning any mRNA they can find, and when they find it, they cut it. So that gets rid of messenger RNA. Now I'm going to tell you, because I like the names, the proteins are called dicer and RISC. Dicer because it dices up the double-stranded RNA, and RISC stands for RNA induced silencing complex. It's actually a complex of proteins. Okay, let's have a look at a worked example then. Imagine there's a gene, we'll call it gene X. A cell needs to produce loads of protein from gene X. So gene X is transcribed and its mRNA hangs around a long time. It's long lasting. Possibly it hangs around for 24 hours before it gets degraded. That means that you can make loads of protein from that without having to regenerate the mRNA over and over again, which requires energy. Just a reminder then that some mRNA will have a half-life of maybe a couple of minutes. So 24 hours is extremely long. But suddenly the cell is told it needs to change function. It mustn't produce protein anymore. Well, the mRNA for gene X is there and will be there for several more hours. So what happens is we transcribe gene Y. This produces a long piece of RNA that folds back on itself. Dicer chops it, and a small part of that RNA binds with RISC and scans the cell for mRNA. Whenever it finds the mRNA, it matches. In this case, gene Y matches a short part of gene X, and gene X's mRNA is cut in half and removed. Very quickly, translation of protein will stop, and this regulation process has therefore prevented the cell from wasting energy.